So, right, we have that recording. So just be aware that the meeting's being recorded. So if you do ask questions and things, that will be on the recording. And without further ado, I'll get started. So just housekeeping, you all know, I've just told you we will record the webinar. This means that anyone who's registered will receive a link to the recording and these slides after the fact. So don't worry too much if there's something you want to jot down or a link in here. You will have access to the slides to be able to grab those things afterwards. There should be lots of time for Q&A at the end, but please do feel free to, jumping forward, to add questions either in the Q&A or the chat during the webinar as they occur to you. It's easier for me to find them if you put them in the Q&A, but whatever works. If I see them during, I'll maybe respond if it's easy. If not, I'll come to them at the end. And as I mentioned, this is very much an introduction. You've obviously found this either through us sending out emails or otherwise, but there are other webinars, different levels, slightly more advanced, or about publishing and getting credit that you can sign up for. If you want a more tailored overview, sign up for a demo and you can get some kind of one-on-one time to talk about how to do the things you're interested in doing. Okay, for today though, I'll start with a bit of context about why sharing methods matters, give you a quick intro to protocols.io, just at the very high level, and then a bit more of an overview of how you can use protocols.io, different ways you can work with the system, and then plenty of time for questions and answers, hopefully. So we'll get started with why sharing methods matters. So most people are researching, and the currency that's understood is publishing research articles. If research articles do not have sufficient information in them, it becomes very hard afterwards when you look back at papers to understand what those experiments were, what the protocols were, how those researchers performed whatever research they performed. And so that leads to a lot of frustration. The tweets I'm showing here, on the left is a biologist who was looking for a protocol in a paper published in 1997, which suggested, as described in another paper from 96, as described in another earlier paper in 87, and maybe that 1987 paper is only available on paper, maybe it's paywall and they just can't access that information. In the middle is a physicist who has hit exactly the same problem. Devices were fabricated as previously described, and then the original reference had devices were fabricated with conventional methods, which really makes it difficult to know what happened and how these things were created. So this leads to a lot of frustration. The GIF in that tweet on the left exemplifies this very nicely, but researchers then can't find, access, or replicate experimental methods and details from research papers. So is it just the research papers that are a problem? There was another initiative, the Biology Reproducibility Project. This was funded and carried out by the Center for Open Science in the States, with a lot of researchers involved. They wanted to repeat all of the experiments from 50 published cancer research papers. So important, impactful cancer research papers that had been published, 50 different papers. When they tried to do this, they found it very, very difficult. They both looked at the papers, but they also went to the original labs and spoke to those researchers and requested the information. Even talking to those researchers, they couldn't get the information about the experiments and protocols that had been performed as a part of those papers. So there's a really nice write-up about this in The Atlantic, which I've got that excerpt from here. The hardest part by far was figuring out exactly what the original labs did. Papers come with methods that theoretically ought to provide recipes for doing the same experiments, but often those recipes are incomplete, missing out important steps, details, or ingredients, and in some cases the recipes aren't described at all. Researchers simply cite an earlier study. So repeating and building on previously published work is extremely hard, but the fact that the researchers too couldn't give the information when asked just tells us that there's a systemic failure to retain the methods information about experiments that are being performed on a daily basis. So this initiative was stopped after they'd attempted actually just 18 of the papers replications. They're planned to do 50 initially, but they reduced to 18 because time and money, it was too expensive, both financially and from a time perspective, trying to get the information to replicate those papers. And in a much more recent study that they put out on that's available on the Center for Open Science website, they actually note that from those 53 papers, 0% of the protocols were completely described. So even for the ones where they managed to get some information, they didn't get them from the papers. But the take home really is that papers and researchers don't have sufficient information for experiments to be repeated. Do we care? Does it matter if we don't have the methods? Methods really do matter for reproducible research. They matter for reanalysis of data also. So the image here are cookies that have all been baked from the same recipe, but with tiny little changes made to the recipe, so small tweaks. So I took this image from a magazine. They were all made by tweaking the same recipe, so you can see where I got this from. But if cookies are your data and your results, and you want to analyze the phenotypic things, the size, the thickness, the texture, any of those elements of these cookies, you can only interpret them in the context of what changes were made to the method. So the tweaks that were made to the method. Maybe it was baking time, maybe it was something that happened in the ingredients and you used slightly different quantities, etc., etc. If you're preserving data and you want to reanalyze them at a later date, you can only do that if you know what the methods were that were used to create the data. And we share a lot of data now. There's been a lot of movement to making sure we share data widely alongside research papers. We really need the methods as well. So there's a wonderful quote from an evolutionary biologist that nothing in biology makes sense except in the light of evolution. I tweak that quote. I say nothing in research data makes sense except in the light of how it was generated. So if you are sharing data, you really need to share methods too. And this is where protocols.io comes in. Our mission is incredibly simple: to make it easy to share method details before, during, and after publication. So if you haven't come across protocols.io and looked at it at all, it is a method sharing platform. It is an open access repository. So if you place protocols onto protocols.io and publish them, they will be available with a CC BY license for anyone else to come and make use of. This landing page, the protocols.io/welcome, is a brilliant place to start if you haven't looked before. You can scroll down and it will give you links to some example protocols and example workspaces. And you could scroll through those and have a look at them because they're open access, so they're available. It has over 14,000 public protocols. And a lot of researchers use it for their method development before they're ready to share more widely, or because they just want to keep things privately within their research group. So there's a lot more content that's private on the platform. A huge number of users, and actually well over 2.5 million views in 2022. So these are views from people who are finding that public content. So it's very useful for discovery and communication of methods scientifically or just in research in general. So if you're using it before and as you go through on your day-to-day, it can help you manage and share research data and protocols. So should those cancer researchers have used protocols.io, they might have much better records of what they did before they published their papers. Has a lot of collaborative functionality. So if you're working within a team to develop methods this way, it's a brilliant way to share the information, tweak, optimize, comment backwards and forwards on methods as you're working on them. And in the grand scheme, if you're doing it this way, it really helps you save time and keep work organized, because if it's time to publish and you need full methods details, if you have them on protocols.io, it's very simple to take the next step to publish the methods that you've put onto the platform. Right, so I'll take you now and show you a little bit of protocols.io. So we're back to that welcome page. This is really easy location to sign up for an account if you haven't already done so. So top right, you'll find a sign up. It will bring you this screen here. You can create an account. You can continue with ORCID if you want, sign in with the Google email. If you already have an account, obviously you can sign in otherwise, but you can link nicely to ORCID and Google and so on. It may be that you're at an organization that has a site license, and I'll talk a little bit in a moment about what the licenses are, but anyone can sign up for a free account. You don't need a license. There are just benefits if you're somewhere that has a license. And that's a place where you can check if you do have or you don't have via this link. Partway down the page, there's a drop down and you'll see the locations that have the licenses. Very, very simple, therefore, to make an account. And anyone can do that for free. Similar important thing here, if you are not interested in making an account, but you want to have a look for content on protocols.io, the search at the top of this welcome page will allow you to search all of the published content. So say you put into their PCR and wanted to search for somebody else's PCR experiments that they've shared and published on the platform. So you'll search PCR. It will bring you to a slightly more advanced search page, and publications will show you any published protocols that have been added to the platform. So you can scroll through those and see if there's something that looks good for you. You can see at a glance the metrics, so how many people have looked at this protocol or downloaded it or bookmarked it. You can check through all the metrics of things like this. If you weren't searching for PCR and you were searching for something a bit more complex, like organoid, because you want to look for a common space, is there a workspace for an area that's open and public where I could try and join and find more information and communicate with other researchers? So if you searched for organoid and then clicked to workspaces, you'll be able to find potentially relevant workspaces. And so there's one there, which is organoid and assembloid. You would have the option to join this. I know it's an open one, so you could join communities that are self-organizing on protocols.io this way and join them. You can look at the publications in there. You can start discussions with other members in these locations if you want. What can you do with the protocols themselves? If you, without an account, you can view them and export a PDF of it to print out if you want, or you can just view it on your screen. If you sign in with an account, you can then also run them dynamically and store run records. You could copy them, fork them, make your own version that you want to make edits to if you want to do so. So that's what you can do with all of that published content very easily without having to do much actual adding of protocols yourself to the platform. Here's just another example of a very open profile that we have, where there's a lot of very brilliant content that sort of self-organized through the pandemic. And you can see this has a large number of members and discussions and publications. I think actually I should update this screenshot because I think there are a lot more in there now. This is a little bit outdated. And then in terms of the basic free account versus the premium accounts, anyone can sign up for the open research free account. You can have up to 5 private files and you can publish for free. So if you create a protocol and then publish and continue to make your protocols publicly available, you'll continue to be able to add more private files, because once they're published, they're no longer considered one of those private files. So it's very easy to do pretty much everything functionally with the open research free account, but it is a little bit limited on the number of files. The premium account has the ability for you to create as many workspaces, have as many private files. You can add up to 10 collaborators into a workspace, even if they're not in the same location as you and don't have that license. You can track full history of changes made. So if you're collaboratively editing and someone makes changes you're not keen on, you can roll it back to an earlier version. You can see some of the history with the basic account, but I think only 10 steps, so it's just a little bit more limited. You will get tech support, obviously, with the free account, but we would obviously prioritize helping people with the licenses if it came down to it. Most of the time, everyone's queries get dealt with very quickly within a day or two anyway, so it's not a slow process. So using protocols.io, so just keep an eye on the time. When you sign up and make an account, you will be prompted to create a workspace. So you'll make your own workspace, perhaps, and then you'll land into the file manager, which is exactly what it sounds like, the location where you can arrange folders and files, place content if you want, set up subfolders to have, published protocols, protocols in development. You can organize your content in here. If you have more than one workspace, they will show up in that left-hand column, and you'll be able to toggle between them and switch from one workspace to another. Most users will not have very many, but it might be that it's useful to have one for a different project, or one for different groups and collaborative efforts that you're working on. And then you can arrange content within the workspaces, but you can also arrange within a single workspace. So that's not too limiting at all. When you've added a protocol, or you create a new protocol, you will come into the editor and the viewer of the protocol or document. You can make documents too. And this is kind of a schema showing you the layout of the editor a little bit, and you'll be able to create nice step-by-step protocols, have sections, add components for variables, volume, temperature, time, scales, etc., and really make nice protocols. And when you're in there, you can toggle very easily between edit mode and view mode. And obviously, if you opened up someone else's protocol, you'll be seeing the view mode, but you won't have the option to edit it. So that's slightly how that works. And then I showed you before the coronavirus method development and the organoid, there's a profile page. So each workspace will have a file manager view and a workspace profile view. And when you create workspaces, you can decide if you want them to be public and visible. That means they'll show up if you do a search, like I showed you earlier, that organoid one, or you want one just for your lab, or you can make it invisible and not public, and no one else will be able to see that. Only you will be able to see what's in there. You can still publish protocols from within that workspace, and they will come up in search results, but you can keep the workspace completely private if you want to. Within that profile, if you do have other members, this is also a location where you can see who the other members are, you can interact with those people, mediate discussions, and so on. Okay, so my computer's going a bit slow. So the file manager I mentioned, this is where you can set up structure, have files to export, run records in development. You can just arrange this however you like to put your content into protocols.io. I mentioned you can see all the other workspaces. I end up in lots because I end up added to many different workspaces. So if I want to, then this is where I can go between the different workspaces. Any file type at all, you can just drag and drop files in here or just upload many things. So if you have a bunch of protocols on your desktop, but you want to put somewhere in a shared location with other people, can be that just using protocols.io for storage of that as a central location is a good way of using the tool as well. You can see with something selected on the right hand side, there's an export option, and actually if I click that through, there are places I could push content to Dropbox or OneDrive, various other things, links with some electronic lab notebooks, etc., etc. And if I click on one of my workspaces on the left-hand side, it opens out this option here. So you can see I'm in a few different workspaces. For my workspace, I can go between the folder, that file manager view here, and the profile, this profile view. So this is kind of how you can navigate and you can access a lot of the administrative things here. There's many other functions and ways of configuring your workspace. I won't go into too many details, but I'll show a little bit in a moment. You can set it up exactly how you want it. So if you have other team members in there, you can control a little bit what capabilities they have within the workspace. So here is what you would see if you click out on administration. You can have access to all of the settings, which is what I'm showing on the right just now, but you can also manage who is a member, invite other people, and so on, and can configure other things here if it's your workspace. When you make the workspace, you're the administrator. You can designate someone else to be a co-administrator, and they would be able to make changes too. So there are many things you can do. If you click to view advanced options, you can change if you want that workspace to be public or private. You can work out how people can be members. So if this one's private and I want to say I will invite people, no one else will be allowed to join. If I clicked by request, people could ask to join, but they wouldn't necessarily automatically be granted that right. I would still be able to choose who could be invited. You can also get a link to send out. And when you do invite people, this is the view that you'll see. You can add in their email address to invite them, or you can just give them that link, and it's very easy to control that way. Step back. Once we've set up a new account, maybe I don't want to collaborate with anybody, but I've made my workspace and now I want to start making protocols. So when you're in the file manager on the right hand side, you'll see a new, and it will give you these options, various options. So if I wanted to upload files, I could find that here too. But we'll say click new protocol. It will ask me where I want to put that protocol. So I could just pick the top level workspace. If I didn't have any subfolders, that's where it would go. If not, I can choose a folder that I've created. I can select what sort of protocol it will be. This will not limit functionality afterwards. It just tries to give me the options that are most likely to be useful to me for that protocol. So if I clicked biology, then I will see all of the biology components on the right hand side. If I click again on these arrows here, then I can toggle between chemistry, computational, or I can just say all if I want to see all of those options on the right hand side. But essentially, you can just start then writing. You can add a title, click new step, and start adding steps to your protocol very, very, very easily. What about if I have a Google Doc or a Word doc or a PDF of a protocol already that I want to put into protocols.io? We do have a mechanism for this. So you can copy paste in. Does need to be checked afterwards by you, especially if it's PDF, because sometimes there are just formatting things. But once you've made that new protocol, you'll see this. If you have an existing protocol, you can copy paste, insert steps from a text file. So if you click that, you'll see this view here. So depending on how you have it in your file, each number is a step, each line is a step. You can choose the correct thing. And you'll do copy from, I'll show you now, I've copied from a Google Doc and then I paste it in. I find it's better to it's jumping through, a right click and a paste and match style, because sometimes if you copy out of Word or a doc like that, there's a lot of tagging that comes with it and you don't want that. But if you do right click and paste and match style, it will come in correctly. So you can see I just pasted in a 16-step Google document, click preview, and then it will give me a preview of how that's then pulled into all of the steps. You can say add to the protocol, and it will pull in all of my steps like this into protocols.io. As I said before, it is then on you to double check things have pulled in and formatting hasn't gone funny. Sometimes, superscript, subscript, or other elements like that might have not pasted in as you would want them. So you do need to just double check. But it's easy enough, you're in the edit mode, to come in, edit anything that needs a tweak. Oops, jumping around. And if you wanted to, because that doesn't give you any of the actual components, if you selected the 5 minutes and then duration, the component on the right hand side, you can format these to take then advantage of the components that we have. 95 degrees C, you can grab the temperature one and format that accordingly, and so on. We also have a protocol import service. I'll come back to this right at the end, but essentially if you have a PDF or a doc, there's a cost with this, but you can submit through this URL here, submit the protocol, and we will import it for that fee. If you're somewhere with a license, you may have a right to this without a fee, but again, that you might need a code for that, but we can send that information if you think that's something you've got a right to. I'll show this page again at the end. In the editor, so this is just showing you a little bit more, very basically, what it looks like when you've made a new protocol. If you're going to do the adding of, you can add a new section at any point in time, and I've just called these section 1 and section 2, but it just to show you that they get color coded very nicely. And they will create a table of contents. So if you have a long and complicated set of experiments with clear subparts, you'll be able to navigate through and use the sections to help you with that. You can see the table of contents on the right. And if you're in the view mode, you'll see a link to the table of contents at the top left. The components are these elements you can add in, which make things much more visible: time, temperature, volumes, all sorts of other things, reagents to point things out. You can concurrently edit. So if I had shared this protocol with a colleague, or they were in this workspace too, and they were sharing and editing it at the same time, you would see their image up here, very much like a Google Doc. When people are concurrently editing, you see that like this, and you get this lovely step-by-step format. So where in a research paper, traditionally, you have a narrative paragraph or two, which doesn't really help for following a method. Protocols.io allows you to show it very much as that step-by-step detailed method, the recipe, if you will, with all of the ingredients and times and conditions and all of that extra information that's helpful. I can see a question about, can Protocols.io replace a supplementary document? I'm going to come to a really good way of how you can link to a Protocols.io method in the context of a research article. So I will answer that in a moment. I'm not ignoring that question. I did spot it. And when you're adding your protocol, we always recommend including as much information as possible. You can add an abstract, you can add an image, which will maybe make it easier for you to find later, for someone else to find and understand at a glance what that is. Add all of the authors. Keywords are very helpful for findability and discoverability later on. And we would recommend spelling out acronyms as well as adding acronyms, so that depending on how you would come back and search later, you would find it with either of those things. You can add a manuscript citation if you have it already. You can add that later as well if you haven't yet published the paper, but you would like to add that in later, it's possible. You can acknowledge your funders and put grant IDs. If you just step between the different tabs that we have, you can add in, safety warnings, specific instruction about before you start, any other relevant guidelines, materials. You can list these all in here, search for all your reagents and create a library of all the materials straight away. If you add a reagent into a step, it will also appear under the materials as well. So you can see that in 2 places. In the context of a research article, for the methods part, you can't generally add figures or videos or like how to or anything that's much more useful for replicating an experiment. On Protocols.io, you can take a photo of the setup, you can take a short video on your phone or a longer video and pop onto Vimeo or YouTube or somewhere for free. And then you can add those into your protocol. So the left one here, that would be insert image or video. That would use if you had a short video on your phone. and it will pull them in. Or you can embed media. So if you've put onto YouTube or somewhere else like this, you can go to YouTube, click the share button that they have there and embed video, copy out this little bit of code, and you would paste that into this spot here, and it will pull the videos into the protocols really easily. So that if it's a tricky step, there's something very peculiar about the setup that it will be helpful to follow, you can share that video as well. So you can put a lot more detailed information to increase the reproducibility into protocols on protocols.io. I mentioned that you can export earlier. This is just to show that in slightly more detail. So either from the editor, you can click more and then export PDF, and then you've got all of these options, or from the file manager, similarly, you have this export on the left with all of these options. The options are the same in either location. And we do have integrations with a couple of electronic lab notebooks. So from within protocols.io, you can push to OSF, the Open Science Framework, where some people have kind of drives for storage of content. Lab archives, more formal electronic lab notebook. You can log into your account there and push content to lab archives that way. We do have integrations with 2 other electronic lab notebooks. Sorry, my slides are clicking forwards. The RSpace and SciNote, but those integrations are from the electronic lab notebook. So if you're logged into your RSpace or SciNote accounts, you can validate into protocols.io and pull content back. So it's a really nice way of, if you're creating the protocols, when you've got the final version, when you want to keep a dated version of what it was like at some particular time, daytime, or if you have run the protocol and kept a record, the run record, which is date time stamped and correlates with data. Remember the recipe tweaks and the cookies, you can have that date time stamped exact record of what you did when you ran the experiment that day and correlate it back with the data. So this allows you to pull content off. Again, if you're limited to 5 files, this can be a good way of you kind of exporting content out to free up space if you need to. Sharing, posting and publishing protocols is really very easy. You've probably noticed on some of my screenshots, there's very prominent share. If you click more, you get these options to post draft, reserve DOI or publish. And if you're in the view mode, you see these very prominently at the top, or under more, there are a lot of other options of the things you can do. It's incredibly easy. If you click to share, you can add, I forget what I have. No, I haven't got. If you click to share, you can share with people by email. You can share with another workspace. If you're working in your workspace, but you're doing a coronavirus protocol, you could share it to that coronavirus methods development community if you were a member of that too. So it's a really nice way of kind of sharing in lots of different locations. Or just privately, my colleagues, their email, I want them to have a look. I can control whether they can view and edit, or if they're a co-administrator. There are lots of ways. The control is all with you. And if you do publish, it's just a kind of 6-step process. It's very, very easy to follow. Protocols are very dynamic. Before publication or after publication, other users can come along and add comments to them. So if you're doing this privately before publishing, and you and a collaborator are trying to get it just right, you can add comments on one another's protocols like this, which will just flag something that needs to be edited, for example, or maybe you're just going to ask a question about comprehension. If it's a published protocol, you can add the comments privately. I think I have another one. Yeah, this is an example on a published protocol. You can see this one has 123 comments, and you can see other users have come and added public questions onto the protocol, and the authors then answer them as well. So if you click that, you'll see when you scroll around, there's a plus that will prompt you if you want to add a question. So you can put a comment or ask a question, type in whatever your question is. By default, they're public. You can ask it privately if you want. We encourage that public, because if you ask a question, someone else might have the same question, and it will save the owners, the authors, time if they only have to answer it once. So on these published protocols, it turns the protocol into having its own kind of public FAQ. The reason these authors, Stephen Henikoff, puts these protocols onto protocols.io is because he was getting so many questions by email that it became a time-saving thing to only answer the questions here, where other people would then have access to those answers as well. Okay, so a question earlier was about, can you replace a supplementary document of the methods section in an article? You can do better than that. If you publish the DOI, if you publish the protocol and get the DOI for it, which is this 6-step process, if you play around, you'll see how easy it is to do. You can grab the DOI, and so this is an example from a PLOS biology paper, and they say methods and protocols for the various different experiments are available as a collection in protocols.io, and then there's the DOI link. So if you click the DOI link, it will bring you to protocols.io, to the version that was available at the time that the paper was published. So here it was a collection. They've actually updated that collection. So I've not talked about versions, but once you've created a protocol and published it on protocols.io, it's very easy to make a new version. If something changes, you get new equipment, you realize something needs to be clarified. And that's what these authors have done. So if you follow the link, you come here, it will tell you there's a newer version. And do you want to see that? So you can click to view the current new version. You'll be able to compare back and see what's been updated. But this gives the authors a lot of control to make new versions. It means that the methods become much more of a living document added on to the paper, but that there's always this dynamic permanence of the link to the version that was there at the time the paper was published. So it's much, much better than supporting information file, because I'll show you in a minute, and as you've seen from search earlier, the protocols themselves are findable by searching on protocols.io. A lot of those over 2.5 million people that came to protocols.io are referred from Google as well. So the methods themselves are independently findable in a way that a supporting information file just would never be. Another option, if you are looking to publish a method, is a new article type that was started by PLOS One called a lab protocol. It's not just restricted to lab protocols. This was the first one, actually, and it's an archaeology imaging technique. And they are proper research articles, but they have to have a protocol on protocols.io, and that essentially replaces the materials and methods. So in the materials and methods, it literally points across to protocols.io, but there's a prominent see the protocol at the top as well. And if you click across to protocols.io, have this bi-directional, if you hover over, you'll see that reference on PLOS One, and you can click backwards and forwards. But then you have a lovely step-by-step method on protocols.io, which goes with this lab protocol article. And it does facilitate cross-disciplinary connections too. This is a lovely example. The top left was a researcher in Chile who was looking for an RNA extraction method from primary cortical neuron cultures, so from some brain cells. After a few tweets, this was a postdoc at UCSF, she suggested a protocol that's on protocols.io, an RNA extraction protocol. When we looked at this protocol, it came from a paper that had been published in GigaScience, which was a method applied to a 3-spine stickleback parasite, so a fish parasite, a fish worm actually. If these authors, these researchers looking for a method for primary cortical neuron cultures had just searched the literature, they would never have found this method in this paper without the method having been shared independently as a complete protocol on protocols.io. So you can increase the discoverability of your methods by having them as separate. And if that had been a supporting information file here, it would have been even less discoverable. So that wouldn't have worked either. You can increase the discoverability and the reproducibility, because chances are this method here will have a lot more information in it than just a narrative piece of text would have had somewhere else without that whole format for step-by-step. So facilitates these disparate and really interesting research connections between different disciplines, even enables that reuse, and actually ultimately enhances the value of the research. You know, this being available means more people will find it and be able to reuse those methods. I am not going to go into it now, but it's a really useful tool, protocols.io, for teaching as well. So you can imagine in computational classes and lab classes, protocols already on for students to use and run, or requesting that students add a method to protocols.io so that they understand how to do that correctly. There's a lot of ways that it can be adapted and is adapted and used by those in different universities for teaching. I showed you how you can add links to papers. Of interest to note that over 500 journals, most publishers, most journals are perfectly happy. And actually, if you look in detail in their instructions to authors, many of them actually encourage using tools like protocols.io to share the methods in their research articles. It's better for them too, because it means the research they're publishing is more replicable and reproducible. So many journals are very happy for you to do that. They don't consider it scooping, it's considered like a preprint. So it's completely fine if the method has been shared before. We have more and more funders as well who require or recommend methods details be shared as a part of the research process and the research that they're funding. And more and more institutions are signing up for these licenses, as I mentioned earlier, that you can check and see if you have that. And just to reiterate that, you can check to see if you go to this protocols.io/institutions, partway down the page, you can check to see if your university or institution shows up as one that has premium, and then it will give you instructions on how to claim that premium account. If you don't have, but you'd like more information or advice on how to advocate for that at your institution, you can contact us on the email shown here. And a very quick recap about the protocol import service, which if you haven't used protocols.io before can be a good way to get started, especially if you have content that you want to get into protocols.io. It can help get you over that activation barrier of starting to use a new tool. You'll submit it. We have two editors, enter and double check, to try to make sure there aren't errors, because this is a manual process. You will then review that. We'll tell you when it's ready for you to take a look at. So you'll need to go through. You can request some edits. We'll come back and make any edits in response to what you've said. And when you're happy, we reassign it to you. So you'll be the owner to share privately, to publish it if you want to do so. You will then, it will be your protocol. So you do need the account, because you need for us to be able to have this back and forth with you and reassign it at the end. But it can be a good way of seeing how what you already are used to working with can be represented on protocols.io. So it's worth thinking about trying. And that brings me to the end of the slides and any Q&A. So please feel free to add questions into the Q&A or in the chat, and I will do what I can. So hopefully I answered, Ade, your question earlier about being able to publish, but I've also popped here my email address, that general support email address for protocols.io, which is monitored across time zones. So is a better way of not having something get stuck in my inbox if you have a specific support question, general support question. And I mentioned at the start, you can also sign up for a demo. So if you want more of a handhold on a specific functionality and to have one of us go through with you, you can book a one-on-one demo. I offer a few slots on a similar time zone to this now, which can be useful, but we have colleagues in America across time zones who offer demos at different times in the day. So whenever suits for you, feel free to sign up. But yeah, that is everything I was going to cover. If you have questions, please do add them now. I will perhaps stop the recording and we can go from there. Okay.